This is one of the online lectures for physiology during week 16, covering broadly reproduction. So the in-person, quote-unquote, Zoom lecture was on reproductive physiology and endocrinology, which tied in with one of the labs that we did during week 16. This is more in line with the lab that we're doing in week 17, focusing on genetics. So when we talk about patterns of inheritance, what we're talking about is that relationship between the DNA that is provided by the egg and by the sperm. So this is inherent to reproduction. And really, when we're thinking about genetics, what we're doing is predicting the possible outcomes of fertilization. So that's why it's so inherently linked to this reproductive unit. Again, I know I'm posting this a bit late, but it does line up a little bit better and more supportive of week 17 anyway, where you're going to get a lot of that practice in lab working through genetics problems. Some monthly learning objectives for this are to distinguish between a genotype and a phenotype. Those are related terms, but they mean something a little bit different in terms of inheritance and the way that the progeny or offspring might look. So we'll be thinking about how to predict those genotypes and phenotypes using different tools such as Punnett squares as well as pedigrees, which are more about what actually happens. So it's kind of like a retrospective looking at patterns versus a Punnett square where you're actually predicting something. So the way that two copies of a gene, one from one parent, one from the other parent, can interact, kind of follow these different patterns of dominance and recessiveness. And if one of those genes is located on what we call an autosome, chromosomes 1 through 22, then it's autosomal. If it's on a sex chromosome, it might be X-linked or Y-linked, but we're looking at conditions that are autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive, and then we'll briefly touch on Y-linkage, but we won't go super into that, especially not for lab. We'll also think about some patterns of inheritance that are not quite as simple as dominant or recessive. We'll look at codominance and incomplete dominance, which we call non-Mendelian patterns of inheritance. So I just wanted to briefly review some stuff that we've talked about over the past couple of lectures, specifically in terms of gametogenesis through the process of meiosis. So remember that when we make gametes, we are forming haploid cells. They only have one full set of chromosomes, but the majority of our cells in our body have two sets of chromosomes, one from one of our parents, one from the other parent. But when those egg stem cells and sperm stem cells begin this process of meiosis, we don't know which copy of a gene is going to end up in that egg or in that sperm. So we kind of have an idea of what's happening through gametogenesis, but we don't know exactly what's getting into that egg or sperm. Remember that the process of mitosis, of making exact copies of cells where your amount of DNA doesn't reduce, happens throughout our life from the time we are single-celled zygotes through adulthood, and it's constantly happening at different rates in different body tissue. When we go through the process of meiosis, that's spermatogenesis in the testes and oogenesis in the ovaries. That process is reducing the total amount of DNA in our cells. Instead of being diploid and having two copies of a chromosome, those cells are then haploid and only have one copy of a chromosome. Again, we don't know if the copy from our mom or our dad biologically is ending up in our cells that are being produced. We just know that there's going to be one of them. So when we go through this process of genetics, we're predicting the likelihood that a certain copy of a gene, or specifically a form of a gene called an allele, might end up in the egg or in the sperm. So to summarize that, genetics fundamentally is a tool to predict the result of fertilization. We don't know what copy of a gene is ending up in an egg that's being produced. We don't know what copy of a gene is ending up in a sperm that is being produced. And then we have the added combination of that egg and sperm coming together and interacting with one another. So we're looking at multiple generations. We're looking at the parents of the individual who's making an egg, the parents of the individual who's making the sperm, the person who makes the egg and the sperm themselves, and then also their potential offspring. So just a few terms that I think are really important to have a solid foundation of going into this lecture include chromosome. I kind of think about this like an entire street. It's a big package of DNA, and there's a lot of stuff going on there. Each street in a residential neighborhood has lots of houses. So each of those houses can be a gene. It's the coding section of DNA. It maybe is a combination of non-coding and coding regions, so introns and exons, but together they make a section of DNA that's going to result in a protein product, the house. A street might have front yards, and it might have roads and sidewalks and different stuff in it that don't actually produce a house, but that gene is specifically a coding section that does make something tangible. So when you're trying to track down a house, you have to know the address. And we can actually look at a location on a chromosome or a locus, which is a specific position on a chromosome to identify a certain gene. So for example, if you have two homologous chromosomes, I'm going to draw them before synthesis, so they look kind of like lines. Let's say that this is chromosome 1 from your mother, maybe your biological mother, and this is chromosome 1 that's being donated from your biological father. If we look at the exact same locus on this homologous pair, so if we go to this address right here and this address right here, we're going to find the exact same gene on those two locations. So for example, that might be a gene that codes for hair color. If we look at a different locus, so for example, if we look down here, we would find the exact same gene on the homologous chromosome at that same location, at that same locus. So that might be the gene for being able to taste a chemical called PTC. Both of those depend on your underlying genes, and both of those have a protein product that is being transcribed by or coded for by whatever gene is at that particular locus. So if we're thinking about those different forms, we might have the exact same gene at a certain location, but we might have different forms of it. So alleles are alternate forms of genes. For example, the house that I grew up in, one of my friends lived around the corner from me. His house was exactly the same as mine. It was just a different color. And we see that a lot in kind of those residential communities, like tract houses, where it's like all from the same builder, and it's just slight variations on a theme. So it's fundamentally the same house, it's just slightly different. For example, in terms of like human traits, my mom is white and my dad is Indian. His family's from India. They both have hair, they both have eyes, but my mom's hair is blonde and my dad's hair is black. My mom's eyes are blue and my dad's eyes are brown. So they have the same general traits, or the same general genes present to code for traits. They just have different forms of those genes. So they have lots of different alleles. So again, alleles are alternate forms of genes. Both homologous chromosomes have the exact same genes. And in fact, if we looked at chromosome 1 in any of us in our classes, we would find the exact same genes, but we would find lots of different forms of those genes. So for example, if we look at Daenerys Targaryen and Khal Drogo, they did not actually have a child on Game of Thrones, but there was a child in a dream that she had. So she has very distinctly blonde or almost white hair. He has very dark brown hair. Her child has brown hair. So they all have genes that code for hair, they clearly have different forms of those genes, and the way that her genes are interacting with his genes is a little bit different. So there's clearly one that's maybe dominant or showing up over the other. But why do some proteins show up over others? There's different relationships between genes and between the proteins coded for by those genes that help us understand which one is actually going to be visible. So I like thinking about this as something in terms of volume. A dominant protein or a dominant trait is going to be what you can actually hear. So for example, if I'm blasting metal music, that's what you're going to be hearing. If I'm also playing classical music, that's only going to really be visible or audible in this case if the metal music isn't there. So one of those is dominant, the metal music. The quiet classical music is recessive. And again, maybe in a gamete, we're looking at those individually, but in our diploid cells, we're looking at them together and seeing how they interact. So we often use specific letters as symbols for dominant and recessive alleles. A capital letter is generally considered dominant, a lowercase letter is generally considered recessive. But sometimes in more advanced classes, we might do a plus and a minus, or we might do a star and then maybe a apostrophe or something. So these are really just, at the end of the day, symbols that kind of indicate to us whether a particular allele is dominant or recessive. So when we're thinking about what we have in our genes, which specific alleles we have, that is our genotype. It's our type of genes. It's what's in your DNA, what alleles do you have for a certain gene. But the protein product that we either see or detect or know is present is our physical type. It's our phenotype. So that's how you look. It's what specific proteins are made and distinctly used from certain combinations of alleles. So we're going to look at specific genotypes and see how those give rise to different phenotypes. So if we end up seeing the dominant trait, we have a couple different options for what's actually in our genes. We can be this big D, or big D, little d. If you are D, big D, that means that you're homozygous dominant. It means you have two copies of this dominant allele, and that's just like blasting a bunch of metal music. That's what you're going to hear. If you blast metal music and classical music, you have one dominant allele and one recessive allele. That's called being heterozygous. So homo means same, hetero means different. So if you're homozygous dominant, you're just homozygous dominant. If you're heterozygous, you're just heterozygous. It's not like heterozygous dominant or heterozygous recessive. Hetero means that you have two different alleles. You have the dominant and the recessive. Here you have two copies of the dominant. So if we see that dominant trait being expressed, we know we have at least one copy of that dominant allele. We don't necessarily know whether we have a dominant allele or a recessive allele for the second one. That pattern emerges when we breed different organisms together and we look at their progeny or their offspring. If you are homozygous recessive, you have two copies of that recessive allele. If we can see that recessive trait, we know for sure that we are homozygous recessive. So we can kind of guess some stuff about our genotype based on our phenotype. If we know our genotype already, we for sure know our phenotype, given this case of simple dominance. So remember, just a quick note that DNA can code for more DNA or it can code for protein. When we go through that process of synthesis and replication in the middle of the cell cycle, that's DNA coding for more DNA. When DNA codes for first messenger RNA and then for protein, that's through transcription and translation, and that's happening throughout the entire cell cycle. So here, just to kind of summarize this idea of gene expression, also called protein synthesis or central dogma, DNA codes for messenger RNA using RNA polymerase. That's an enzyme that polymerizes RNA. It puts together a big messenger RNA molecule through a process of transcription. Translation then is using that messenger RNA to assemble a protein, it's a code for a protein, and that's done using ribosomes. The way I think about this is your DNA is kind of like a big cookbook that's in every single one of your cells. If you just want to jot down a single recipe from that cookbook, that's like transcription, and then translation is like actually cooking from that recipe. So when we use these arrows here, it does not mean that DNA is turning into messenger RNA. It means that DNA acts as a code for messenger RNA. But it does show us that there is an inherent relationship between what's in our genes, that existing underlying code, and the actual proteins that we see, the traits that we have. So before we get much further, I just wanted to say that generally, when we're thinking about proteins or traits or phenotypes, we have a tendency to think about stuff that we can actually observe and see. But keep in mind, you have so many proteins in your body. For example, you have genes that code for lactase enzyme. That's your genotype, and then having your lactase enzyme is your phenotype. So there's kind of different regulations or controls of transcription. We might have the gene for something, but we don't end up assembling the protein. We might have different forms of genes and then get different forms of the protein. And those proteins can be things we see, like eye color or hair color. They can also be things we can't see, like the ability to break down lactose, having the lactase enzyme. There's also a chemical called PTC, phenylthiocarbamide. It's found in Brussels sprouts and different cruciferous vegetables that makes them really bitter. About 3/4 of us have the ability to taste that because we make a certain protein receptor that is found on our taste buds. The other 1/4 of us actually can't taste it at all. So that protein may be something visible, and a lot of the time it often is not. So again, genetics fundamentally predicts the results of fertilization. Those gametes, the egg and sperm, are haploid, and they come together in fertilization to create a new diploid offspring. So when we solve problems in genetics, we're making haploid gametes, and then we are kind of predicting the results of those gametes coming together in diploid offspring. So when we talk about simple dominance or these simple genetic patterns, we call them Mendelian traits. The reason for that is this guy named Gregor Mendel, who you can kind of see in the background of this slide. He was a monk back in the 1800s, and back then monks really just brewed beer or worked in gardens or did illuminated manuscripts, so kind of living the life, just a super chill kind of academic existence with a lot of beer, which is kind of inherent to academia if you know any professors. But anyway, he was really passionate about his education. His family actually gave away his sister's dowry in the pursuit of getting him his education, and it was a big thing to him. So when he was out there with plants, he started to kind of breed different pea plants together, and he noticed that there were some patterns in the traits that he would see. So back then, people just thought that the traits of one parent and the other get blended together to create the offspring. But he noticed that some of these traits were skipping generations, or there was some interesting stuff happening with the math. So he actually really carefully bred them together in specific ratios and combinations and created true breeding lines that he could then kind of cross together intentionally and record his results. So he used quantitative or number-based data to show how different traits were passed through the generations. And the traits that followed really simple patterns were called Mendelian. So based on Gregor Mendel, he kind of got lucky in that he didn't have anything weird like codominance or incomplete dominance. It was just, he was observing traits that happened to follow simple dominance. So he came up with these two laws, the law of segregation and the law of independent assortment, and these are genetics laws, but you can see how directly they link to the key events of meiosis. So remember, meiosis fundamentally forms haploid gametes. That's basically what the law of segregation says. When we have chromosomes going into those gametes, the chromosomes from that individual's mom don't all stick together. The chromosomes from that individual's dad don't all stick together. They get mixed up in different ways. So those chromosomes go into gametes independently of one another during meiosis, and that is basically Mendel's law of independent assortment. So again, there's a fundamental relationship between genetics and meiosis. So I'm sure a lot of you have done Punnett squares in Bio 5, but also in school, probably in middle school and high school. So these Punnett squares are fundamentally a biological tool to predict the genotypes and phenotypes of offspring from different sets of parents. So when we're looking at this, right here, these are the parents, the P generation. These are the ones who are making eggs and sperm. And we see that they're diploid. This male has 2 copies of this Z chromosome, or this Z gene. This female has a Z and a W. So again, these are just symbols. This could also be like big D, and big D, little d. It's just this particular example is showing sex determination of sex chromosomes in a bird. Don't worry about that for now. So when we're thinking about what sperm are produced, for example, with this, because he's homozygous dominant, we know that both of the sperm that are capable of being produced are going to have that dominant allele in them. But with this one over here, when the eggs are produced, if we just have an egg, we don't know whether it has the Z gene or the W gene in it. So there's a 50% of that being the Z gene. Oh gosh, sorry. And there is a 50% chance of it being the W gene. And it would be the same if we were looking at those symbols that we saw before, a 50% chance of it being a big D and a 50% chance of it being a little d. So we also don't know which sperm is going to end up fertilizing which egg, so it's basically a numbers game. We draw the alleles within their gametes along the outside edges of the Punnett square. So these sperm go down here. It doesn't really matter if you do male on the top and female on the side or vice versa. This is just how we're doing it here. And then these possible eggs go right here. So then we bring this sperm and this egg together and we see what they make. We put this sperm and this egg together and we see what they make, and so on. So I'm sure a lot of us have that experience of just randomly putting together these letters but without the biological context. So it's important to remember what this is actually telling you about haploid gametes and diploid offspring. So this is a tool to predict the possible progeny or offspring produced by fertilization between a sperm and an egg. Pedigrees are another biological tool that we use again retrospectively. So these are a way to represent a family tree. Sometimes we might, generally they show biological sex. So if they're male or female, they often show whether they're affected by a particular condition, and then they sometimes show whether an individual is deceased or whether they are a carrier, meaning if the disease is recessive, they might be a carrier if they're kind of carrying it in their alleles, but we don't see it. So if they're heterozygous, they're called a carrier when the condition is recessive. There are some limitations here. So for example, with this particular pedigree, I went through and I did all the calculations, and you can't tell based on the information provided to you by this pedigree if a particular condition is autosomal recessive or autosomal dominant. So that's kind of a limitation of pedigrees because it's retrospective, it's not predicting, you're only as good as the data that you have available to you. So some patterns just aren't going to emerge in nature. So again, we know this pedigree shows an autosomal disease because there's equal ratios of it happening in males and females. It's not sex-linked, but we really can't figure out whether it's dominant or recessive. So when we're looking at what we can actually observe, we can calculate something called the phenotype ratio. That is the number of offspring expressing the dominant trait to the number of offspring expressing the recessive trait. This kind of tells us something about fractions or proportions, which are actually out of a total, but the ratio is just number to number. So for example, if a family has 3 brunettes and 1 blonde, their phenotype ratio might be 3 to 1. And when I say 2, I am meaning like TO, not TWO. That's what that little colon is telling us. So the phenotype ratio might be 3 to 1. The likelihood or the proportion of being, for example, brunette in that family is 3 out of the total, which is 3 plus 1, which is 3/4 or 75%. So a 3 to 1 ratio is the way to say the phenotype ratio. If you're asking about the likelihood or the proportion, then you might use these values. Genotype ratio is something that we often have to infer or assume if we have pure breeding. So pure breeding means that we have generations and generations of individuals who are all homozygous dominant or all homozygous recessive. We know there's not some combination, some heterozygosity happening. So the genotype ratio is looking at the kind of ratios within the progeny of particular genotypes. And here, because there's not just 2 options, we're considering whether they're homozygous dominant, homozygous recessive, or in between heterozygous. So in that previous example where we had 3 brunettes and 1 blonde, we know that blonde is going to be, sorry, I'm losing track of where my pen is. We know that blonde is going to be homozygous recessive. But when they are brunette, we have no way of knowing whether they're homozygous dominant or heterozygous, and if it's if it's simple dominance. So just assume simple dominance for this. So here, those 3 might be any combination of those genotype ratios. However, if we know we have 1 homozygous dominant, 2 heterozygotes, and 1 homozygous recessive, then we can get that same exact phenotype ratio from our genotype data, because this is going to add up to 3 dominant to 1 recessive. So if we actually apply what we were just talking about in the context of a Punnett square, generating the Punnett square and then solving it without actually drawing in all of those gametes and zygotes, we kind of get what we traditionally think about with a Punnett square. So for this first generation, this is kind of like the parent generation. In this example, you have an individual who is homozygous dominant being crossed with an individual who is homozygous recessive. So this is what we're talking about with these true breeding or pure breeding lines, homozygous dominant and homozygous recessive. So when we make that cross, just to kind of briefly think back to what we were just talking about a couple slides ago, let's say that this is the male and this is the female. This A right here, this capital A, represents a dominant allele being inside of a sperm, and this represents the other possible sperm. This represents an egg, and this represents another egg. And then these are going to represent potential diploid zygotes. So the offspring that are possible from these crosses. So because you have two pure breeding lines, all of the resulting potential diploid zygotes are going to be heterozygotes. They're going to get one dominant allele from one parent and one recessive allele from another parent. So when we're looking at these, a cross between homozygous dominant and homozygous recessive results in all heterozygotes. And if this is a case of simple dominance, they're all going to express that dominant allele. That's what we're going to see, and they're going to have that dominant phenotype. So it results in a 4-to-0 phenotype ratio. There's 4 individuals that have that dominant trait being expressed and none that have the recessive trait. And because we know something about the parents, we can say that it also has a 4-to-0 genotype ratio. All of the resulting offspring are heterozygotes, and then there's none that are anything else. And really what this should actually be is 0 to 4-to-0. So there's no homozygous dominant, there's no homozygous recessive, there's just heterozygotes for the genotype. So let's say that we take 2 of those resulting heterozygotes, 2 of these potential offspring, and then we cross them together. That's not so common in animals, but it's quite common in plants to cross 2 individuals of the same generation from the same parents. So if we cross 2 heterozygotes together, we get something a little bit more interesting happening. Now we have a case where there's a couple different types of gametes that can be formed from each parent. So we end up with 1 individual who is homozygous dominant, 2 individuals who are heterozygotes, and 1 individual who is homozygous recessive. So when we look at those, it turns out that these 3, the homozygous dominant and the heterozygotes, all of whom have at least 1 dominant allele, are going to express that dominant phenotype. So we end up with 3 individuals with that, of the total or possible, who have that dominant phenotype for what in terms of what we're actually seeing. And then there's going to be 1 individual down here who has the recessive phenotype. So if we're looking at the phenotype ratio, we have 3 dominant and 1 recessive. Sorry, that yellow is a little bit hard to see. And then for the genotype ratio, we have 1 homozygous dominant, we have 2 heterozygotes, and then we have 1 homozygous recessive. So when we cross 2 heterozygotes together, this is what we call a monohybrid cross, that crossing of 2 heterozygotes. So we get this kind of classical 3-to-1 phenotype ratio and 1-to 2-to-1 genotype ratio, or 75% dominant phenotype, 25% recessive phenotype. So when we start to look at those patterns, if we were talking specifically about the dominant allele in that situation, maybe it codes for a particular disease. That disease would follow a pattern of inheritance that we call autosomal dominant. It's equally likely to happen in males and females because this allele that codes for this particular disease is not located on a sex chromosome. It's located on autosomes, which is a fancy way of saying non-sex chromosomes, or chromosomes 1 through 22. So if a gene is located on an autosome and it is seen when it has that dominant allele, then it's autosomal dominant. So that dominant allele is expressed when the genotype is homozygous dominant or heterozygous. You just need at least 1 dominant allele in order to express this characteristic. So these conditions don't skip generations. You can always see them. Oftentimes we think about diseases as being autosomal recessive because there's kind of a selective pressure against them. If a disease is dominant, it's quite obvious, and often that affects reproductive rates. But if it's like a novel mutation that causes a dominant disease, then there might not be any way to avoid it. So for example, achondroplasia or dwarfism is caused by an autosomal dominant condition. There's another one called neurofibromatosis type 1. That one causes tumor formation in the nervous system and leads to skin and skeletal deformities. That one, if you have even one copy of that allele, that's what you're going to express, this condition. For autosomal recessive, and sorry, I forgot to animate that arrow correctly, but this is also located on an autosome, but in this situation, it's coded for by a recessive allele. So you only see it when the genotype is homozygous recessive. So if we look at this pedigree that tracks different biological sexes, and in this case, it's showing us the genotype as well as the phenotype. So in this example, we also have to take into consideration carriers. If an individual is heterozygous, then they only have, let's see, in this pedigree, I don't like it when they do this, but they're drawing it so that the blue is unaffected, meaning that they don't have the disease. So this boy right here is homozygous dominant. This carrier daughter has one copy of that affected allele, the recessive allele that causes the disease. Same with this carrier son over here. And then the affected daughter has two copies. She's homozygous recessive. But because these can be masked by being a carrier, you're going to end up with having to keep track of those individuals that are carrying that recessive allele in their genotype. So one common example of this is cystic fibrosis. It's caused by an autosomal recessive, it is an autosomal recessive condition. It's caused by an allele that is on an autosome, and you have to have two copies of it in order to have cystic fibrosis. Other ones include sickle cell anemia. With sickle cell, if you have two copies of the dominant allele, that's just normal hemoglobin. If you have one copy of the dominant allele and one copy of the recessive allele, you have protection against malaria. And if you have two copies of the recessive allele, you have sickle cell anemia. So that is another one where those recessive alleles are kind of hiding in the population. Having just one copy of them confers evolutionary advantage, but having two copies is quite dangerous. Tay-Sachs disease is another one. That one is very common in Ashkenazi Jewish populations. If you've ever heard of the Ashkenazi panel, that is a genetic test that people who are part of the Ashkenazi Jewish community have to take before they end up having children, because there's like a pressure to intermarry with people of a similar background from you, some of these recessive alleles are brought together more often than is kind of what you would expect to see in the whole population. So when you have people who are breeding together in close communities over extended periods of time, you have higher incidence of these autosomal recessive conditions. So it's not just the Ashkenazi Jewish community. Anytime you have a religious community within a larger population where there's a pressure to intermarry with people from a similar background, it's likely to happen. We see this with migration patterns too, for example, Cajuns in Canada and the United States, so different conditions like that. And then phenylketonuria is another example of an autosomal recessive condition. So I just kind of went into a spiel about this, but why might affected individuals be more common in isolated populations? Because you're more likely to have these carriers mating with one another instead of mating with someone who's homozygous dominant. If there's any of those recessive alleles, they'll be brought together more and more often in the population. So just as a reminder, we talked about eggs and sperm. And when we talk about eggs and sperm, these are sex cells. They are fundamentally cells. They have lots of genetic information in them. When we're talking about sex chromosomes, that is very different. So sex chromosomes are specifically the X and Y chromosome. a lot of the time we have X and X coming together to be the sex determinant for females. X and Y come together to be the sex determinant for males, specifically because the Y chromosome has a gene on it called the SRY gene. In those cases, that SRY gene, when activated, is associated with producing testosterone, with gonadal tissue developing as testes, and with your ductal system forming. Keep in mind that sometimes the SRY gene gets kind of cut out and moved onto an X chromosome, in which case you have an XX male. Sometimes it's mutated or missing or inactivated or doesn't get switched on. In that case, you would have an XY female. So again, just having the gene does not mean that your chromosomes are a certain way. It doesn't mean that your sex determination is going to play out a certain way. It's just kind of on average what happens. So there's a fundamental difference between your sex cells. Eggs are generally produced by people who are XX, but not always, and sperm are generally produced by people who are XY, but again, not always. So there's your overall sex, there's your hormones, there's your anatomy, there's sex cells, which are those gametes that are produced through meiosis, and then there's the sex chromosomes, which are involved in sex determination. So it's really important to think about whether we're talking about a gamete, like an egg or a sperm, versus a chromosome, which is a package of DNA inside of that egg or sperm. Those sex chromosomes, again, have genes that help determine secondary sex characteristics, which we talked about extensively in the other 2 lectures for this week. So if a gene is located on a sex chromosome, it's not going to be equally likely to be found in males and females as determined by sex chromosomes. So there's going to be some unique genetic patterns that we start seeing, especially in terms of how 2 copies of a gene, 2 different alleles, interact with one another. So this image on the left is showing, it's a color blindness test. It has a red spotted background, and then there's a green 5 in the middle. That red-green differentiation is hard to distinguish if you are red-green colorblind. And red-green colorblindness is determined as a sex-linked condition. So the gene that codes for colorblindness is located on the X chromosome. Remember that females tend to have 2 X chromosomes, males tend to have an X and a Y. So here it's showing you that it's X-linked recessive. So we're tracking the X chromosome of the mother, who has 2 copies of it. The father has 1 X chromosome and 1 Y chromosome. We see that the Y chromosome is not carrying that allele because it's X-linked. It's located on the X chromosome. And so we can't just draw this Punnett square as C and then, or big C and then big C and little c. We have to also track what chromosome those are on. That also helps us determine the chromosomal sex of the offspring. So when we look at sex-linked traits, we have to track those sex chromosomes in addition to whatever allele we're looking at. So we're looking at 2 different things now. So we draw those Punnett squares a little bit differently for sex-linked traits. This example of an unaffected father and a carrier mother is exactly what we saw on the previous slide, except this time it's looking at a different X-linked recessive condition called hemophilia. So hemophilia, we'll get back to in just a moment, but is basically associated with not being able to clot correctly. So we'll look at this X-linked recessive in a little bit more detail over the next few slides, and I will also help you draw this out. So again, we have to track those sex chromosomes. We have to draw out the X and the Y in addition to the alleles for a particular disorder. In this example, capital H is the dominant allele and lowercase h is the recessive allele associated with hemophilia. Again, those alleles are traveling on the sex chromosomes, so we draw out those sex chromosomes and then we draw those those different conditions as superscripts next to each letter that represents a chromosome. So I'm going to go ahead and draw this out and kind of explain how we get here. So if we're looking at that same example that we saw in the previous slide, the mother was a carrier. So she has 2 X chromosomes. She's chromosomally female. She's a carrier, which remember means she's a heterozygote. And when we say heterozygote, we're talking about the alleles of those genes that are traveling on the X chromosomes. So she has 1 dominant and 1 recessive. This guy is actually traveling on that X chromosome. And something that I think is kind of confusing is the letter X looks a lot like the way that a chromosome is drawn. Here we're really talking about specific X chromosomes. So she is going to mate, which unfortunately is also demonstrated using a little X, which I'm sorry about. Biology is super confusing sometimes. She's going to mate with a male who has his own X chromosome, and he also has a Y chromosome. Remember that that Y chromosome usually has the SRY gene on it, which is why he's associated with being male. He is unaffected by this condition. He does not have hemophilia, which is an X-linked recessive condition. So we know that he is carrying a dominant allele on his X chromosome. So these guys are going to make gametes, and we don't know what gametes they're making. So she's going to make 2 possible eggs. He's going to make 2 possible sperm. This chromosome with the allele that's hooked onto it, this dominant H, capital H allele that's hooked onto this X chromosome, is going to end up in this possible egg. This guy with the lowercase recessive allele is going to end up in this egg. This one's going to end up in this sperm. And this Y chromosome that's not carrying any of these alleles because that allele is not Y associated is going to end up in this sperm. So then we're going to do our little Punnett square. And I wish I had a stylus to make this less ugly, but I'm sorry about that. So let's just go ahead and draw the female down here. So this is going to be female. And then up here is going to be the male. So one possible egg is going to be this X chromosome with a dominant allele on it. The other possible egg is going to be another X chromosome with a recessive allele on it. Up here, this one possible sperm is going to have an X chromosome with a dominant allele on it. And the other possible sperm is going to just be a Y. So again, it's X-linked. That means that this Y chromosome is not carrying any of this particular allele on it. So when we make these combinations of egg and sperm, this potential child is a daughter according to the chromosomes, and she's not going to be affected by this condition. She has 2 copies of that dominant allele, so she's solid. She does not have the disease. This daughter down here, we know she's a daughter because she has 2 of those X chromosomes, so according to her chromosomes, she's female. She's going to have 1 copy of the dominant allele coming from dad, and then 1 copy of the recessive allele coming from mom. Remember, that's what we call a carrier. She is not expressing this condition, she's just hiding it in her sex chromosomes. So now these guys over here, when we bring this potential egg and this potential sperm together, we have an X and a Y. That means we're only going to get 1 copy of this allele, and in order to not be affected, it has to be that dominant allele. So for this one, yeah, there's going to be the dominant allele, So we have a male that is unaffected. Remember that we have two daughters that are unaffected. Neither of these people have hemophilia. It's just that one of them is a carrier. Her children might have hemophilia. Over here, this first male does not have hemophilia. He has the dominant allele, so it's a recessive condition. He does not have hemophilia. But this male right here gets a Y chromosome from dad, and then the recessive allele on the X chromosome from mom. So here we see that 0 daughters have the disease, but half of the sons have the disease. So as we go through and track these chromosomes with these alleles on them, we start to see differences in incidence and patterns emerging based on sex. When we look at a pedigree and we see those differences, that's the first indication that it might be a sex-linked trait. So it's really easy to think that because a condition is more common in males, that it's automatically Y-linked. But remember, males are unique in that they only have one copy of the X chromosome. So with X-linked dominant, it's a little bit different. It's kind of hard to tell the difference between this and autosomes because you just need one copy of the allele in order to have this condition. So it's not necessarily going to be more common in males. It might be more common in females, but it's sometimes hard to tell. Because females have 2 X chromosomes, they can have a condition in which they have maybe 2 of these or even just 1. With males, they have to get in at least 1, but it's just different amounts of this allele coming in on the X chromosome, and they just need at least 1 dominant allele. So this condition where they're female and have homozygous dominant, that would result in the condition. If they're female and they're heterozygous, that would result in the condition. And then males who just have 1 copy of that dominant allele would have the condition. So we see this in this example with flies. So the red eyes in fruit flies is an X-linked dominant condition. Vitamin D resistant rickets is one example in humans. But when we're talking about especially kind of detrimental human conditions, the one we refer to most is probably X-linked recessive. So when we're looking at those X-linkage patterns, again with X-linked dominant, if we're in a situation where we have an affected father, that's going to result in unaffected sons and affected daughters. So again, this might, with X-linked dominant, it might be a little bit more common in daughters. So with X-linked dominant, if there's a heterozygous mother, then that's going to result in 50% affected sons and 50% affected daughters. So it kind of depends on what the chromosomal sex of the parent is, because that's going to determine and whether they're affected or not. That's going to determine their likelihood of passing it on, and the ratios that you see based on sex in the offspring. So again, when we're thinking about these crosses and specifically with sex linkage, recessive conditions are the ones that are the most commonly talked about and often the ones that have more or higher incidence in chromosomal males. So here, that causative allele is located on a sex chromosome, specifically the X chromosome, and the causative agent is the recessive allele. So the genotype has to be, if they're chromosomal female, they have to be homozygous recessive. If they're chromosomal male, they only need 1 copy of that recessive allele in order to get this condition. So we talked about color blindness already. We see this Punnett square that we saw earlier, And this is the pedigree version of that same cross. A father that is unaffected, a carrier mother, 50% of sons end up getting affected, and 0% of daughters get affected. So we saw that with hemophilia with the same genotype cross. Here we're seeing it with color blindness. Like I mentioned, hemophilia is another X-linked recessive condition. It's caused when you have mutations in certain clotting factors. So if you fall and you get a bruise even, or you get a cut, then you're just not able to clot and heal that correctly, and you end up hemorrhaging quite severely. So again, when you have these populations where people are breeding together, over time, you end up getting a lot more of these recessive conditions, including X-linked recessive. And so the Russian royal family is kind of a recent example from the 20th century. So the family of Tsar Nicholas II, we see that his son Alexis had hemophilia. And then if we look at a pedigree of all royal families since Princess Victoria and Edward Duke, see a lot of these people, royals, if you think about like blue-blooded royals, they all interbreed together because they want their children to marry other royalty. So you end up with these families who have these alleles being carried and hidden in certain generations and then expressed in others. The Y-linked condition is not dominant or recessive. There's, again, only going to be situations in which there's one copy of the gene. So for X-linkage, there are situations in which there's only one copy because of chromosomal males, but there are also conditions in which there's two copies with chromosomal females. So we can't determine whether they're recessive. If there's only one copy ever, we can't determine if it's dominant or recessive because there's nothing to compare it to. So in Y linkage, it's just going to be found in every single male. So if a male has a Y-linked trait, if they get that Y chromosome from their parent who has the trait, then they're always going to have that condition. There's very rare situations in which you can be XYY. That's called super male. Then I guess in those situations we could see and compare between those two. But usually this is from chromosomal non-disjunction, so those are going to have the same genotype anyway. So again, Y-linked conditions are only found in chromosomal males. One kind of interesting pattern of inheritance that is non-Mendelian is codominance. So this is a situation where you have two separate alleles that are expressed equally. One is not dominant over the other. You can see and visualize both of them perfectly well. So one example of this in flowers might be if you have a splotchy flower that has two separate colors, like if it's like red and white in equal amounts, that might be codominance. In a human example, blood type is the most common. So if you're type A, you only have the dominant A allele. You might have the recessive O allele. If you're type B, you might be homozygous dominant for B, and then you might also be heterozygous with O. But if you're type AB, if you have an allele that codes for the A antigen on the surface of your red blood cells and an allele that codes for the B antigen, one is not going to be dominant over the other. You're going to have both the A and B antigen on the surface of your red blood cells, and you will not have any antibodies circulating in your plasma against those. If you're type O, you just straight up do not have any antigens on the surface of your red blood cell in terms of ABO blood typing. With incomplete dominance, this is what people used to think was the case for everything, this kind of blending of patterns or colors. So in this example with flowers, there's the red flowers and the white flowers together, they make pink. It's just a blending of traits. So it occurs when neither allele is dominant. So the previous one was codominance. Both of them are going to show up equally. In this one, none is really going to dominate over the other. They're both going to kind of blend together if you're heterozygote. So you get a unique blended phenotype. And there's not a ton of examples in humans, but some of the most common are hair pattern. So it's a little bit more complicated and nuanced than this, and if you have curls, you know that it is something that changes over time, and your phenotype is very flexible and hard to predict sometimes. I have very curly hair, I just straighten it. But with your hair pattern, oftentimes if one parent has very curly hair, another parent has straight hair, you might get wavy hair in the offspring. So there's some blending of traits happening there. With eye color, you might also see this where you end up getting hazel. So here's just kind of a summary slide of the different pedigrees. On Canvas, you can access this and kind of spend some more time looking through it. Again, with the autosomal recessive and the X-linked recessive, you see a lot more skipping of generations. A couple of reminders, make sure that you take that quiz that's available this week. Keep an eye out for your reflections on Canvas. There's one due in the next few days that's going to be worth 20 points, so please make sure you get that done. There's no new lectures this week, but you do have lab. There's a genetics lab, and you should prepare a draft of your term paper, which is due Wednesday, May 13th for both sections. The next required Zoom session for lecture specifically is if you're in the Monday class, Monday, May 11th. If you're in the Friday class, it's Friday, May 15th, but you do still have lab sessions. For my Monday class, I'm recording this Sunday night, and I'll send you a reminder email that Monday morning during our normal class time, it's not a mandatory Zoom session, but I am going to be helping you guys with genetics for lab if you want